Glycolysis is the first process that occurs within glucose metabolism. Glycolysis is a very important mechanism, and if we understand step by step what is happening, we will be able to retain this information for a longer period of time and truly appreciate what's happening in this process. So essentially, the purpose of glycolysis is to break down glucose to yield energy. But how do we initiate that process of glucose breakdown? Well, we have this glucose molecule, and our biological systems have a mechanism in place in which we phosphorylate this glucose molecule to signal to the cell that it is ready to undergo glycolysis and glucose metabolism. So when our energy levels are low in a biological system, enzymes will phosphorylate the glucose to signal to the cell that, hey, break this down because we need energy. And that's the first step of glycolysis. It's to phosphorylate glucose to signal to the cell to break it down. This step is catalyzed by the enzyme hexokinase. And we have magnesium 2+ as a cofactor, which isn't going to be important right now. It's essentially just needed to balance out the charges. That's why we have that magnesium. So how do we phosphorylate glucose? Well, we need a phosphate group. How are we going to get that phosphate group? We're going to get it from ATP. So ATP stands for adenosine triphosphate. So it has 3 phosphate groups. We're going to take one of them, put it on our glucose molecule, and we're going to be left with adenosine diphosphate, so 2 phosphates. So on our carbon number 6, we are going to phosphorylate with a phosphate group. So now we have glucose 6-phosphate. Remember, it is still glucose. We just say 6-phosphate because we phosphorylated on that 6th carbon. Now, what's important to note here is that the delta G is negative 16.7, meaning this is an exergonic process. And our KEQ is very large, and since it's very large, it means that the products are favored. But if we were to look at this reaction in isolation, if we were to just look at glucose to glucose 6-phosphate, this would not be an exergonic process. It would actually be endergonic, and the value is plus 13.8 kilojoules per mole. So if we look at the conversion of just glucose plus phosphate to glucose 6-phosphate, it's endergonic. So where are we getting the energy to catalyze this reaction? We are getting it from the hydrolysis of this ATP molecule. When the ATP is broken down to yield that phosphate in ADP, the energy released is negative 30.5 kilojoules per mole. And this is for the ATP hydrolysis. So since the hydrolysis produces negative 30.5 kilojoules per mole of energy, that is more than enough energy to allow this process to occur. So essentially what's happening is that this breakdown occurs, we get that production of energy, that energy is inputted for this process to move forward, because this process requires an input of 13.8 kilojoules per mole of energy. So the first step, phosphorylating glucose to signal to the cell to break it down. We need ATP because we want to phosphorylate. We need phosphate from somewhere, and we're getting it from ATP, and this reaction is exergonic because of that ATP hydrolysis. Now in step 2, it undergoes an isomerization reaction because glucose 6-phosphate is of no use to us. We want the isomer of glucose 6-phosphate, which is fructose 6-phosphate. Since these two are isomers, it means the chemical formula is the same, just the chemical structure is different. So the glucose 6-phosphate is an aldose, whereas the fructose 6-phosphate is a ketose. Now, this isomerization reaction is catalyzed by the enzyme known as phosphohexoisomerase. I forgot to mention something before in the previous step, but in the previous step, the enzyme catalyzing the reaction was hexokinase. And whenever we see a kinase, that means ATP or ADP is involved. Okay? But that was a hexokinase because we were phosphorylating our glucose. But now we are looking at an isomerization reaction, so our enzyme is going to be an isomerase, specifically a phosphohexoisomerase. Why? Because we have a phosphate group and our sugar is a hexose. We have 6 carbons, so it's a hexose. Phosphohexose isomerase will catalyze this conversion from glucose 6-phosphate to fructose 6-phosphate. Now, since the KEQ is really small, it means that our reactants are favored, and this is slightly endergonic. Not highly endergonic, it's only 1.7. Now, in step 3 of our reaction, this is the second phosphorylation. So in glycolysis, it's important to remember that there's 2 phosphorylation steps. Step 1 we discussed was the activation. Now, step 3 is our second phosphorylation. Where is that phosphorylation going to occur? It's occurring on carbon number 1 of fructose 6-phosphate. So now we'll have fructose 1,6-bis phosphate. Bis just means 2. So we have 2 phosphates. And we see that we have 1 phosphate here, 1 phosphate here. Where are the phosphates located? On carbon 1 and carbon 6. What molecule do we have? We have fructose. Now, this process is catalyzed by the enzyme phosphofructokinase, or PFK1. Once again, we have a kinase, which means ATP or ADP will be involved. And we see that because, remember, we need phosphate. And where are we getting it from in step 3? We're getting it from ATP. Once again, the hydrolysis of ATP will give us energy, and that will contribute to the exergonic nature of this step. Since the KEQ is very large, once again, it means the products are favored, so this will be a spontaneous step, and we will get energy. Now, once we have our fructose 1,6-bisphosphate, step number 4 is to undergo an aldol cleavage, which essentially means to split the molecule in half. When we split it in half, we'll get 2 molecules. We'll get dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. This reaction is catalyzed via the enzyme aldolase. And what's important to note is the delta G is very high, 23.8, so it's highly endergonic. Our KEQ is very small, which means the reactants are strongly favored. So how do we get this reaction to move forward if it's so endergonic? The reason why this reaction is possible is because the concentration of glyceraldehyde 3-phosphate is kept, it's always kept low. It's kept low because steps 5 through 10 are actively using up product. So essentially, if we are using up product in the steps to come, what's going to happen in step number 4? Well, what do we know about Le Chatelier's principle? In his principle, he says that if you add a stress to an equilibrium, the equilibrium will shift to relieve that stress. So if the concentration of glyceraldehyde 3-phosphate is always kept low, the equilibrium will shift to relieve that stress. So that's how we get this reaction to move forward. We always keep this concentration low in glycolysis in order to pull this reaction forward. And this is referred to as product pull. In step number 5, we have a triose phosphate isomerization. So remember, in our previous step, we produced dihydroxyacetone and glyceraldehyde 3-phosphate. We're okay with the G3P, but we do not want this dihydroxyacetone phosphate. We want to convert it into something that is actually usable for glycolysis. So we convert it into another G3P molecule. We do that through the enzyme triose phosphate isomerase. Triose because 3 carbons, phosphate because we have a phosphate, and isomerase because, remember, these 2 molecules are isomers of one another. They have the same chemical formula but different chemical structure. This step is a little endergonic and it has a small KEQ, not too small, so the reactants are a little bit more favored. So Here it is important to note that we've produced another G3P. So now, here on forward, in glycolysis, we have two G3Ps, so steps 6 through 10 will occur twice because we have two G3P molecules. Now over here, step number 6 is an oxidation reaction where we have inorganic phosphate and it is being added to our G3P molecule. It's being added onto this carbon over here. Now both this carbon and this carbon have a phosphate. This process is slightly endergonic. It is small KEQ, so reactants are a little bit more favored. This reaction is catalyzed by glyceraldehyde 3-phosphate dehydrogenase. Dehydrogenase, important enzyme to make note of here, because whenever we see a dehydrogenase, NAD plus NADH is involved. So in this step, we are losing a hydrogen. We're losing this hydrogen over here, and NAD plus becomes NADH. And whenever we have NADH, we'll always see free proton. So essentially what's happening, oxidation reaction, oxidation referring, so let's go back to this. Leo says GER. Well, oxidation is loss of electron, and we can see that we're losing our electron, which is our proton. So we lose this electron, we get NADH, phosphate group attaches, we get 1,3-bisphosphoglycerate. Remember, bis means 2. What do we have 2 of? We have 2 phosphates. And this molecule is referred to as glycerate now instead of glyceraldehyde. This is very important to note here, the nomenclature. So we have 1,3-bisphosphoglycerate. We've made NADH, and this is the enzyme that catalyzed. Step number 7 is substrate level phosphorylation. This step is exergonic. It is being pulled forward, spontaneous. What's happening? We are getting our first ATP molecule. So this phosphate that we just attached in the previous step is going to be broken, and that phosphate is going to attach to an ADP molecule, and we're going to form ATP. So here it's important to note that we have 1,3-bisphosphoglycerate. We removed that phosphate group on the first carbon, so now it's just 3-phosphoglycerate. No longer it will say bis. Now, remember, we're coming back to our kinase. We have phosphoglycerate kinase. Phosphoglycerate, it's in the name, and kinase, because why? We are dealing with an ADP or ATP molecule. Whenever we see kinase, think of ATP or ADP. KEQ is very large, so we know products are favored. Spontaneous reaction, we have Mg2 plus as a cofactor to balance the charges. Step number 8. We have our 3-phosphoglycerate, and we're gonna undergo a little bit of a rearrangement, but this is a specific kind of rearrangement because we're not forming an isomer. We're using a mutase, which is it's kind of a type of an isomer. So essentially what's happening is that one of our functional groups is being transferred to another location. So we have this phosphate group. It's not happy over here. We want to get rid of it, and it's more energetically favorable for us to move it over to carbon number 2 so we can break it and attach it to an ATP. So we use a phosphoglycerate mutase enzyme. It's in the name, phosphoglycerate. Mutase refers to the movement of a functional group to another location. Slightly endergonic process and small KEQ, but it's not too small, so reactants are favored, but not by that much. And since our phosphoglycerate is now on carbon number 2, we change that number to reflect the molecule. Step number 9, a dehydration. Whenever you see an enolase, think of a dehydration. This is the enzyme catalyzing this reaction. Where are we getting this water from? We're getting it from right over here. What happens when we get rid of these atoms? These 2 carbons will form a double bond because remember, carbon wants to have 4 bonds. Now this was 2-phosphoglycerate, but now this molecule is called phosphoenol pyruvate. We refer to this in short form as PEP, PEP. This is a very high energy molecule, very important molecule, and this will drive our next step. Since it has a lot of energy, our next step will be highly exergonic. This step, however, slightly endergonic, small KEQ. Once again, reactants are a little favored, but not by that much. Whenever we undergo a dehydration, the purpose of having a dehydration reaction is to disperse our energy, to create more of a high energy molecule to drive a specific process that we want. So this dehydration is occurring to create our last ATP molecule. And we see that over here. Phosphoenol pyruvate, we remove that phosphate, ADP becomes ATP. What enzyme are we going to see? We're going to see a kinase, because whenever we think about ATP or ADP, we're going to need a kinase. We're using pyruvate kinase because we are making pyruvate. Mg2 plus and K plus are cofactors that will just balance the charges in this reaction, which we don't need to know much detail about right now. Delta G, highly exergonic because this is a high energy molecule. KEQ is very large. That means products are favored. And now we've created pyruvate. So our pyruvate will continue on and it will undergo pyruvate processing and it will enter the TCA cycle. Quickly to mention, when we create pyruvate, there are two forms of pyruvate that can occur, enol and keto form. Keto form will predominate in our solution. So to summarize, let's just quickly say what the overall is in our glycolysis. We used 2 ATP molecules. Which steps did we use them in? We used them in steps 1 and 3. We produced 4 ATP molecules. Why? Because we have 2 G3Ps. Each G3P produces 2 ATP. In which steps? Steps 7 and 10. We also produced 2 pyruvate molecules, remember, because we have 2 G3Ps, and we produced 2 NADH molecules. So overall, since we used 2 ATP and we produced ATP, overall we produced 2 ATP. Our net is 2 ATP, 2 pyruvate, and 2 NADH. And this is glycolysis.